Okay, so I'm delighted that Pat Fields has agreed to be our speaker today. I got to know him several years ago when I stumbled across some plant fossils up in McCall and asked around, does anybody already know about this? And they put me in touch with, oh, there's already this expert on Western Miocene flora who spends a month or so each year, summer at the College of Idaho, where he's a senior curator in the museum over in the basement of Boone Hall. And I said, well, that sounds like the right person to link up with. So that led to getting Pat Fields dragged into the Miocene flora, or the new flora up there in McCall, as well as getting me more interested into what had always been a latent interest in paleobotany of the West. So, yeah, I've been happy to get to know Pat. Apparently, he finished his master's degree at Berkeley 2 years before I arrived. He worked on the Payette Flora as a paleoplant ecologist, apparently. And the Payette Flora a lot around Horseshoe Bend, where he had, I guess, some relatives in the area that made that possible for him. And got him started at College of Idaho as well. I mean, the museum there as well. One of the people who's been there the longest. He then ended up getting a Ph.D. at Michigan State University on the Sugar Creek flora. He's the expert on the Sugar Creek flora. Research emphasis on Miocene fossil plants of the Pacific Northwest. He ended up getting his day job at Olivet College in Michigan, teaching biology, field botany, environmental science, geology, physical, geography, and service learning. Just retired in the last couple of years. One of his other expertise, he gives lectures on chocolate as Doc Chalk, not to be missed if you ever have the chance. Does adult education, continuing education courses. Served on the, he's also involved in the Michigan Botanical Club and the nonprofit Michigan Botanical Foundation. We're delighted that even though he's still Michigan based, he still managed to spend as much time as possible with us here in Idaho. And I will now stop jabbering and turn the time over to Pat to tell us about Miocene flora of the greater Idaho region. Thank you, Pat.
All right, thank you for that introduction. I'm going to see if I can manage to do this correctly. First of all, thank you for the invitation and thank you for the introduction. And what I want to do is share with you something about what the fossil plants in Idaho have been like about 15 million years ago. You guys know a great deal about the modern flora, but we thought we'd just take a trip back in time. So I just said, imagine a time when Idaho was covered with deciduous broadleaf forests containing plants like alders, beeches, chestnuts, elms, magnolias, maples, sycamores, but also had things like tree ferns, redwoods, dawn redwoods, bald cypress, avocados, cigar box tree, ginkgos, Oreopanax, sweet gum, and many more. So there's a couple of key parameters to just get in mind, put in the back of your head to set the stage. One is on the left-hand side, we know that vegetation is clearly linked to precipitation. So in the dry deserty parts, you don't get forests. This is, one of those, real, gee, you think. So in the mountains, we get the forest. Why I stress forests is most of our fossil plants that are preserved are forest plants. So that's why there's an emphasis there. And then on the right, we can look at density. And again, it correlates beautifully to where the mountains are, where it's cooler and wetter. And finally, on the right hand, the fourth Idaho over, we can see that forests are named by the dominant or predominant plant type in that group. Then one other parameter to keep in mind is time, geologic time. And so one person envisioned it, I like this, as time just keeps spinning along. I don't know, can you see my cursor? Do people see the cursor? We can see okay, yes. Okay, great. And so by our best guesstimate, the age of the Earth, it was formed about 4.6 billion years ago. And in less than a billion years, and this isn't to scale, so I'm just going to pick a spot. At 3.85, we see the first evidence of life. We see fossils that look like little bacteria preserved in silica deposits. And then when we come up to 1.5 billion, we start to see the first evidence of photosynthesis. So some of these bacteria-like things start to look like algal threads and so on, but also there was free oxygen because all of the rocks prior to that were jet black. And all of a sudden we start to get brown soils. We start to see iron oxide or rust going in there, and that could only have happened if there was free oxygen. If we zoom up to about 700 million years ago, we start to see the first animals, but it's not until right here at this part of the cycle, almost the last cycle and a half or 2 cycles worth, do we see animals with the first abundant hard parts. And so that's when we see a lot of fossils. And that's what helps us understand things. If we zoom up a little later to the Ordovician Devonian periods here, we start to see the first land plants. And it isn't until after the big Permo-Triassic extinction right here at 250 million, after that, do we see the first dinosaurs at roughly 225 million. And then we jump up here to around 130, 140 million, and we see the first fossil Angiosperms, flowering plants. And then there's the big Cretaceous tertiary extinction, where the last of all the dinosaurs except birds became extinct. I don't know if you know, but birds are actually dinosaurs. They are modern dinosaurs. And then we zoom up here to this little pie piece at wedge from about 23 to about 5 million years ago. And that's the Miocene. That's the interval that I'm going to be talking about. And then finally, in about the last 2 or so million years is when we had the most recent rounds of glaciation. And then humans that really looked and behaved like humans didn't even come about till about 130,000 years ago. So that's a quick overview of time. I want show it to you one other way. And that is, here's a cartoon of the sequence of rocks. And again, this base of the Cambrian is when we first get abundant hard parts. So all the stuff I talked about before that is not really taking up much space on this diagram. And then after that, we have eras, Cenozoic, Mesozoic, Paleozoic. We have periods, and they come up here on the right, Tertiary and Quaternary. And then finally, in the Cenozoic, in the Tertiary, we can subdivide them into epochs. So the last 65 million years, we have the best fossil record and the most information. One quick thing to bear in mind, if we were to compare all this to 24 hours of time or 2 cycles on a 12-hour clock face, this Precambrian interview is 1 complete cycle plus 10.5 hours of the second cycle. So in other words, if we compared it to 24 hours, the first 22.5 are all before we have abundant fossils until about 10:30 p.m. And then from 10:30 to maybe 11:20 p.m., we have the Paleozoic, where we see life become more complex and then march onto land, both plants and animals. In the Mesozoic, from maybe 11:20 to about 11:45, is the age of dinosaurs. That's that Triassic, Jurassic, Cretaceous interval. And finally, this Cenozoic period or era, and the Tertiary period in particular, is this tiny little wedge from maybe 1145 to 1159, 58. That's the Tertiary. And then the Quaternary is just the last couple of minutes. And then humankind's time on Earth is just the last few seconds of that 24-hour clock. So I'm talking about a very little tiny interval of time, 23 to 5 million here in this Tertiary period. How is that expressed in Idaho? Here's a geologic map. And if you look here, coming down the 4th block of the colored blocks is Tertiary sediments. Those are the things that contain our fossils. Tertiary volcanic rocks and basalts are the things that weather and get incorporated in and retain our fossils. So it's primarily this sort of pale green. And if we look on Idaho's geologic map, we see that the south side of the Snake River Plain over here is some, there's a little bit over in Sucker Creek, there's some in the Greater Treasure Valley, basically, then Weiser area, a little bit north. And then if we scooch on up to northern Idaho, there's selected deposits. And then in a few other places in eastern Idaho, there's some. That's all the area we have with these tertiary sediments that are loaded with the fossils I'm going to be talking about. One big picture story to put in the back of your mind, finally, the last big picture story to put in the back of your mind, is all of Western North America is basically straddling two big, great tectonic plates. The bulk of the continent is sitting on what is called the North American plate, and it's moving westward. The bulk of the Pacific Ocean, plus a few little slivers like San Francisco and so on, are sitting on the Pacific plate, and they're moving eastward, and these two are colliding. And there's the little tiny Juan de Fuca plate in between. And what's happening is, since the North American plate is made up of a lot of less dense rocks, it tends to get shoved up and over, or conversely, the Juan de Fuca and Pacific plates tend to get subducted underneath this. So in side view, we can see the tiny little thin layer of the Pacific Ocean here. And then the Juan de Fuca plate is going down because the North American plate, it's colliding, and the North American plate is shoving on top. We're seeing accreted terrains, little bits of new land added all the time. And as this plate goes deeper and deeper, there's greater heat and pressure, and so it melts. And the less dense parts rise up, and that's the source of all the volcanism in the Western US. It's also the source of all the igneous intrusive rocks like granites, which if they'd spritzed onto the surface, they'd be lavas and ashes. But since they're stopped just below the surface and spend a few million years cooling, they grow beautiful crystals. So that's the setting. And then there's one other factor. Down deep below, there is a persistent hotspot. We don't quite understand what or how they are working, but they sit still. And as this North American plate gets shoved to the west over them, we see a series of progressively younger volcanic deposits. So how is that expressed? If we look over in the left, what do we see? Here's Idaho in the middle with Oregon, Nevada, Utah, Wyoming, and Montana all the way around, and Washington. I forgot that, sorry. We see volcanic deposits that start roughly 16 million years, and they get younger as you go towards Yellowstone. And that hot spot, referred to as the Yellowstone hot spot, is currently underneath Yellowstone, and it last erupted roughly 600,000 years ago. And then if we put in the Columbia River basalts, we now know the Steens basalt is part of that sequence, all this gray in the right-hand half of the screen. And it started just about 17 million years ago, and roughly from maybe 16.5 to about 15.7, in those 800,000 years, about 80% of all these lavas spritzed out. Those are the lavas, the ashes, the basalts that are accumulating the deposits I'm talking about. So Idaho has this very complex geologic history, as well as interesting climate. So how do we find these, or what do we see? This map shows a whole bunch of Miocene floras that I plotted about 25 years ago. And with the exception of a couple right here in southwestern Montana and a couple in Colorado that are a little bit older that I put in there for comparison in a discussion I was having with this chart, all the rest of these are Miocene. And in the intervening 25 years, many new localities have been discovered. And The other, many more no doubt will be found. And then this red dot, red star is meant to highlight number 25, and it's these 3 dots. That's the Sucker Creek area that I studied for my doctoral dissertation, so I know it the best. And I wanted to show you, here's a blow up map. This vertical line right through the middle is the Idaho-Oregon state line. Idaho's on the right, Oregon's on the left. All of these big round dots, there's 50 of them, represent over 100 plant localities that I have found or have recorded that other people have found. And I've been to virtually all of them now to see what kind of fossils were in there and understand better the greater Sucker Creek flora. Many of you know Fossil Hill. I'm going to talk about that in just a second. But the punchline here is that each of these dots on the map could represent as many as 100 individual localities. So we actually have quite a bit of a fossil record, even though it's scattered in just a few little areas. Many of you might have been to the famous Fossil Hill. This is a view looking westward. There's Fossil Hill. Here's the dirt road, but this right here is the drainage canal. And it goes off to the north and east and brings water to an agricultural area. So that's sort of a reference landmark. This is Fossil Hill. And most people think that's Sucker Creek, that's it. But it turns out there's many other places. Some of them are right down in the creek. And this was a rare interval in 1988 when they actually needed to do maintenance on the dam for Sucker Creek Reservoir. So they let the creek go dry. And it was a rare opportunity for me to get out there with a couple of buddies and start digging like crazy and actually sample what leaves were in these shales that usually are under anywhere from 6 inches to 2 feet of running water. So this was a very rare event. Elsewhere in Idaho, we see places where there are leaf-bearing shales right on the edge of water. But more typically, they look like this. This is yet another place. And there's yours truly sitting there. And all of these, we're up a side canyon to a greater valley. And all of these layers are just loaded with fossil plants. You know, sometimes you won't find anything in a given layer, but you go up a little bit higher, a little bit lower, you find new things. If we jump back one last time to Sucker Creek itself, the near ground is an ash. And that's this ash you see off in the distance. But if we look down here, the creek, Sucker Creek, runs right down through here. These shales and all of this are loaded with fossils. And then we come up to an ash layer. This is the lower ash of 2 biggies there. And then in between it is more sedimentary rock that's loaded with fossils. Then there's the resistant cliff building type ash, and then even more fossils. And off in the distance, this we refer to as the arrowhead section, has even more. So there's actually, once you get out there and start looking, there are fossils almost everywhere, it seems like to me. This is that arrowhead section. I just wanted to give you a sense of the size of it. Down low, about where I was standing when I took this picture, is a really fun fossil flora. No one's a Sucker Creek locality that no one's ever been to except myself and one other person. And then if you look carefully, see all these little marks on the side of this dirt, this shales, and they come up here. This is me sampling about every 6 to 8 inches, up, 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 keep going, keep going, keep going. I finally stopped when I got up here and found good redwood leaves. But the point is, there are fossils at sporadic intervals through all of this. These are lake deposits. This is the bottom third of the arrowhead. There's the middle third. Now I want to just jump to the top. And again, way up here, this is looking the other direction and back down. So this is a view west and southwest. And you can see there are layers. And if you scrape a little bit, pretty soon you find new fossils. So Idaho, actually, once you cue in on the environment of deposition, Idaho has lots of fossil deposits, and they're mostly not yet studied. So we collect these fossils, we put them in wooden trays, we trim them, store them, We identify them. Once we have good identifications, they tend to get put into these acid-free cardboard trays. And then my question is, what's next? What do you do with these things? They sit in a museum. This is the Smith Museum, by the way. This is Janie Burns and Amanda Grant, who is an interpretive ranger at Ponderosa State Park, who came to check them out since some of the fossils near the park are there. So first of all, we find that when you dig up fossils, they come in many different isolated organs like leaves, seeds. I like these two in side by side because this letter B is a 1934 drawing, and here is a scanning electron micrograph of that same kind of specimen. So you really see quite a bit of difference from good old-fashioned drawings up to actual images. All this in the lower right are pollen and pollinomorphs, pollen and spores. They're just about everywhere there's sedimentary deposits, you're going to find pollen and spores. We also occasionally find fossil wood. Very rarely we find fossil flowers, and even more rarely still, we actually find fruit attached to the same two leaves. And then we can say for a fact that the thing that made this type of leaf made that type of acorn, in this case of an oak leaf. Okay, so let's look at some of these things. What we have is assorted lower vascular plants, ferns, gymnosperms, and ginkgo, conifers and ginkgo. We have dicotyledonous angiosperms. Very rarely we do have monocotyledonous angiosperms, although as pollen, we find them a little more abundantly. So now I've whetted your appetite, I hope. Now let's look at things. These are horsetails, equisetum, now classified that whole group as ferns. But we see here's from Pickett Creek and here's from Sucker Creek. I tried to put where they're from in here so that you can get a sense of some of the diversity of places. Here are some regular, more traditional ferns, an osmunda and a thalipteris, both from Sucker Creek. Then there's this really cool thing. It's based on one specimen, but we have no reason to believe there won't be more. This thing right here on the left, letter A, is actually the actual fossil that has been lifted out of the rock from up at Clarkia. And those brown or black dots are the sporangia. And if we look through progressively more detailed images till we get to scanning electron micrographs, you can actually see the trilete spores. All of that provided enough information that they were able to figure out that this is the genus Colcita in the Colcitaceae. And so what I love about this is the image that Idaho had tree ferns 15 million years ago. In the redwood group that's now lumped into the juniper family, we have Glyptostrobus, and here the three pictures on the left are cones. We also have a cone attached to foliage and then foliage itself. So you can see the diversity of this foliage. It looks sort of juniper-like, but not quite the same. And then over on the right, it turns out there's juvenile foliage. And Glyptostrobus, I've seen modern plants of this in the first 7 years or so of their life, make foliage that only looks like the plant on the right. And consequently, you're tempted to say, I don't know, is that a redwood or something? But it turns out it has some distinct structures that show that it's really Glyptostrobus. And with time, these mature leaf by leaf, layer by layer into these more scale-like leaves. There's also dawn redwood, which you may know as a planted tree in yards with the beautiful opposite foliage, opposite leaves. And then it has female cones with the cone scales that are in opposite pairs. And it even has male cones we've found. There's also redwoods that are very closely allied to the coast redwood. And we have cones and we have foliage. And the definitive thing here that you just can't miss is, see this lower half of this specimen? It's redwood foliage, and then it ends 1 year's growth and has a couple of scale-like leaves and then starts the next year's growth. And then way up towards the top of the tree, you start to get more scale-like leaves anyway. These are all just classic redwood situations, redwood examples. We also have bald cypress, things like you see from Florida through Louisiana on up the Mississippi to the southernmost part of Illinois. Bald cypress grew in Idaho in the past. In the juniper family proper, there's tons of things, but I just picked these two to whet your appetite. You may know incense cedar, and this is Calocedrus, and we have foliage and cones. You may also know arbor vitae, often called the tree of life. And this is Thuya, and we have foliage and cones. And these two sets of foliage look similar until you start really looking in detail and you realize they actually can be distinguished, not because the fact that one preserved darker than the other, but the pattern of the leaf arrangement is a little different. And so we can tell these guys apart. The last gymnosperm I wanted to show you is ginkgo. And ginkgo is usually fairly rare, but every once in a while you'll find a fossil flora that's just got it in oodles of specimens. And it turns out this ponderosa flora near McCall that Barbara and I have been working on, and Chris Davidson have been working on, has a fair amount of ginkgo, and that's kind of exciting. In the pine family, there are many, many different genera, but I decided to pick a couple of really closely related things to show you. Pines themselves, and you may know there's the white pine group where the needles look like they're sitting on top of the bundle, on top of a little nub. And what it is they have a basal fascicle sheath that's deciduous. So at maturity, it peels off and blows away. While in the yellow pine group, these needles are held together by a persistent basal fascicle sheath. So we have white pines and yellow pines, and then even within yellow pines, we have at least a couple of different species of pine cones. We also have some white pine cones and tons of seeds and things as well. In the angiosperm world, there are a zillion things. It's really quite fun and it's never ending. We're always finding new things. I did want to point out this one specimen here that superficially looks like a beech leaf, but it's got a fruit attached by a peduncle to the petiole of the leaf. This is an extinct genus in this family, and it was named pseudophagus or false phagus. Most of the rest of these things I'm going to talk about as we move along. So we have liquid ambar. Sweet gum is the modern one. And it looks crudely like a maple or a sycamore leaf, except it's got tons of little teeth all along the margins. And just like in modern liquid ambar, you can find it in a bunch of different leaf morphologies. We even have fruit in various states of preservation. We also have this unique thing that is based so far on one specimen, but the venation is just a dead ringer for the modern American species of smoke bush, smoke tree. You may know the smoke tree that people plant in their yards. That's actually the European one. There's only two species, and the European one's planted all over America. And I've seen it in Idaho. I've seen it where I live in Michigan. I've seen it all over. And then down in the southeastern US, there is this guy, Cotinus ob ovatus, and our specimen looks just like that. I couldn't believe it when I was trying to figure out what this was, and all of a sudden it hit me, this venation, these secondaries come out and always seem to fork at the margin, and then they just sort of disappear into the margin. I said, that's a Anacardiaceae is the family. I said, that's an anacardiaceous feature. And I said, you know what, I was thinking to myself, sitting down there in the Smith Museum in Caldwell, Idaho, saying, this looks like smoke tree. And I quickly ran upstairs and plucked a few leaves off of a tree right on the edge of planting outside of the building and brought them down and said, bingo. And then I've spent the last 5 years trying to talk myself into it being something else. And nope, it keeps coming back beautifully to the genus Cotinus. Also in that same family, we have sumac or rus. You may know sumac as a bushy thing that grows. I see it along highways where I live. And in the autumn, it turns this beautiful burnt red. It goes, they say the sumac's on fire, and then it goes yellow, and then it goes brown, and then they all fall off. And there's also poison ivy or poison oak. Now that thing And this I thought looked like a maple leaf at first, but then I realized, no, actually with the help of a Jack Wolf, a famous paleobotanist, said, nope, it's a tough one, but it's not magnolia, it's poison ivy. And I tried really hard to make him wrong, but he was dead right. He had it beautifully. This thing looks like the terminal leaflet off of a poison ivy or poison oak. We have ilex or holly, and there are at least 2 species. One of them is very deeply dissected, so the width of the leaf across the midrib is very little compared to the length of the lobes. And the other is sort of the reverse of that, very shallowly dissected, or relatively speaking, short lobes. We have this interesting thing, Oreopanax. It's an araliaceous thing that today has living descendants that are in Central America and on into Northern South America. And when it was first found, and I put both these images of the same specimen in here to show you because they highlight different features, but when it was first found, this was thought to be a red or a black oak. And then they realized it's part of a compound leaf with 5 leaflets in this case, 1 little one, 3 big ones, another little one. We now have them with 7 or even 9 leaflets, but Oreopanax is this thing. And so that's a Central American type influence that was living up in Idaho. In the Oregon grape group of things, we have 6 different species. I put in 3 to show the range of variation. We have one that's an ovoid leaf with nice wicked teeth on it. We have one that's an ovoid leaf that's almost entire margin. There's just a hint of some teeth. And then we have one that's deeply dissected and kind of spinos. And there are 3 other morphs that have different names as well. Alders are based on, for the Miocene record of Idaho, 2 different species of leaves, and then lots of female strobili or female cones, if you want to call them that. There's also seeds of alder. And then in that same birch family, there are various kinds of birch, as well as even Austria hop hornbeam. And again, this is another one of those specimens that's been lifted up off the rock and has actually been preserved or stored between 2 sheets of plastic. And this photo was taken by holding it up to a window and just taking a picture. So it's pretty neat, all the detail you can see on the actual fossil in some specimens if they're well preserved. There's hop tree or Telia. This is a tree with 3 leaflets, and the leaflets usually are disintegrated, but every once in a while we'll find a beat up one. But we find a lot of these fruit, and they look sort of like an elm seed with a wing that goes all the way around, or 2 wings and 1 central nutlet, but it has a long beak to it. And we find them in various stages of degradation. But there's also this thing. It was mixed in with those for the longest time. And then people realize this is actually a pair of nutlets sitting between the big wings that go all the way around. And then there's another wing that rides up between them as almost like a saddle over a horse. So it has a small set of wings or bract that rides on both sides, and then the big pair that all surround this pair of nutlets. There is an Asian genus today called Dipelta. So this was named Diplodipelta. We have viburnum. You may know that as a plant that people plant around their yards. The one where I grow that's so common is called nannyberry. You may know Cercidophyllum or Katsura tree. This is a fun one. It's Asian. And what I like is it turns yellow in the autumn, and then the leaves start smelling like this mixture between vanilla and pineapple and banana. It's really kind of fun to smell the leaves of this tree in autumn, and then it turns bright yellow and they all fall off because it's deciduous. In this same family is something that I think this is correctly identified, but I keep flipping in my mind. We have this specimen that seems to compare quite well to something called Trocodendroides. Oides means look like, and there is a family Trochodendraceae in a genus Trocodendron. And this kind of sort of looks like that group, but it also has some other features that compare it better to the tree we were just looking at, Cercidophyllum. So I'll get to Trochodendraceae in just a minute, but we have this interesting thing of still not pegged down 100%. There are at least 2 species of dogwoods, cornus, a narrow one and a broad one. These are both displayed at exactly the same scale. And what you see is the narrow one has secondary veins that kind of are sinuous as they come off of the midrib. And it's for its length, it's pretty narrow. And then we have a broad, more round, much bigger leaf with nice arcuate secondary veins that come off of the midrib. So there's 2 species of dogwood at least. I've often asked, why aren't there more? Because where I live, there's like 5 or 6 shrubby dogwoods, all but one of which is a shrub, and then the other one's the Cornus Florida, the one that's a tree understory. But I'm surprised we don't see more species of dogwood, quite frankly. There's this thing, if you know the Ericaceae, the heath family in California, there's something called madrone trees. They're really quite pretty. And this is that genus, but it compares more favorably to the Irish strawberry tree. So I call it the fossil strawberry tree rather than a fossil madrone. There are lots of legumes. These are terrible pictures, but they do show the feature I wanted to show, which is in here in 3 different morphologies. You can see at the base of the petiole on these, those little ribbed structures. That's a pulvinus. And a pulvinus is an organ that is made of tissue that can absorb water and expand and become very rigid and stout. And so the leaves can be held out in the sun. And then at nighttime for many of these, and typically also when the wind really picks up and it gets blowing, they pull the water out of those little cells and the leaves drop down. And so the pulvinus is kind of like a functional hinge. And you say, well, maybe you're pushing it saying 3, but we have at least 3 types of seed pods. We have Cercis or redbud type. We have Cladrastis or yellow wood type. And there's even Sophora or necklace pod or Kauai type pods found in the Miocene of Idaho. In the oak family, there's many, many, many things, and they're all kind of interesting and challenging. The 2 on the left are a part and a counterpart of a great big chestnut. And this leaf is, if you look at the scale, this is metric. So every 2.5 of those is an inch. This is about a foot long. And then elsewhere in other floras, this was from Paddock Valley, this is from Clarkia way up north. We see this again and again. There's a great big chestnut from Idaho. We also see little tiny beech leaves with very minimal teeth and rarely even beech nuts. So this is the genus Fagus. There are many things that look like oaks or mixed up with oaks have been called oaks. This is one. I think it compares most favorably to Chinquapin, which is a genus Chrysolepis, more common in China. And one way to tell is when you look at these secondaries, they always seem to loop and close. There's a prominent looping that's known as Brachydodromus venation. And what people noticed was, wait a minute, there's a second layer of this Brachydodromy stacked between those big prominent secondary looping veins and the margin. This is known as festooned Brachydodromy, and no Quercus does that. So this was formerly called Quercus, but it's really Chrysolepis. There are a fair number of lobed oaks, like 5 or 6 species. They're both in the red-black group, and in the white group. White oaks are lobed, but without a tooth or a prickle at the end of the lobe. Red and black oaks have that tooth or prickle. So we have lots of lobed oaks. If we get to the live oak group, this is a fun one. This is called Quercus pollardiana, and it has toothed leaves and entire margin leaves. And it seems to compare beautifully to the Canyon live oak, the modern living one that's in the west face of the Sierra Nevadas. And a very rare find was to find this oak leaf, one of these, attached to a stem with 3 intact acorns. This was so good that this specimen was illustrated in a paleobotany textbook. Then I found one day this specimen, which is Quercus simulata, and it has a great deal of leaf variation. It can be entire, it can be toothed, etc. But I found a couple of leaves of it attached to a stem with 3 acorn cups and 2 acorns still present. And I thought, hey, this is wonderful. We can finally tell which of these 2 live oaks makes which acorn. Unfortunately, if you go back and forth to those acorns, they're virtually identical. Can't tell them apart. And yet this tree, the canyon live oak type ancestor, does not seem to make very many leaves that look at all like the Quercus simulata type. This does lead to something I want to share with you, and there are many groups that have their own problems, but this is the biggie for me. Quercus simulata, in its range of leaf morphology, makes leaves that can overlap with all these different species and these genera in their range of leaf morphology. So not only do we find a willow that seems to range and look like Quercus simulata, some forms of it, but we see Castanopsis and Chrysolepis, we see oaks, we see what used to be called lithocarpus, this is now called lithocarpus, various oaks, a chestnut, and all of these can make leaves that look like some of Quercus simulata. The asterisk indicates those specimens, those species that are recognized from Sucker Creek, Flora, and it's really quite diverse and very frustrating. And I've tried very hard to decide, is Quercus simulata an artificial group and it's really all these other things or whatever, but nope, it seems legit. One last point to make is this is not implying a biological connection to any of these. It's simply overlapping range of morphology. So I'm not saying willows are at all related to oaks. I think Walter Beckler would be upset if I was saying that. We do have gooseberries or currants, the ribes, there are several species. We do have even hydrangea. You may know this with the 4 big colorful bracts people plant in their yards, and they often have either pink or white or blue flowers. And then after they go to seed in the fertile specimens, you can actually get these whole collections of capsules. So we have the fruit as well as the bracts, and very rarely, but we do have the leaves. We also have sassafras. You may know sassafras tea or root beer. The root that root beer was first made from was the root of sassafras. Eventually, they figured out it had some toxic compounds, so they generally make it synthetically these days. But we do have sassafras, and we have the specimens with 1 lobe, specimens with, or excuse me, no lobes, 1 lobe, or 2 lobes. So you can find all of those. These were just pretty and photogenic, so I put these 2 in. There's tulip tree, in particularly the Floras to the north. And so we have this, if you know Liriodendron, the tulip tree. I was taught if you walk into a forest in Michigan and you're looking for a tree that would be appropriate for the mast of a ship, a tulip tree will stand right out as the most beautiful straight thing and be perfect. We also have, here's a central axis around which a lot of seeds were formed after it flowered, and all of them have fallen out except the last couple of pair are there. There's magnolias of various kinds, and magnolia fruit from Clarkia, and even here's a modern one to compare, and they really do compare quite favorably. There are lots of other laurels and things as well. I'm just hitting some highlights. I mentioned cigar box tree or cedrella. Here's the capsules, we have the leaves, and we have the seeds. And Murphy's Law, I couldn't find any good photogenic seeds of it, so I stole some from an older Flora, so forgive me for cheating. These aren't Miocene, but the seeds of this look just like that. We have Comptonia or sweet fern, as well as some things, Myrica and the Myricaceae, and they seem to grade, so that needs to be sorted out. But we have those in lots of floras. We have sycamore in quite a few places, and you may know the great big lobed ones that crudely look like a maple, and you can find them with 5 lobes, with 3 lobes, or with only no lobes in particular. We can even find the stipules, the leaf-like structures found at the base of the petiole. And then on the fruit side of magnolia or of platanus, sycamore, we find big naked receptacles after the seeds are fallen out. We even have an immature receptacle with a halo of the seeds around it. This one I don't ever think matured, so they didn't fall off. And then here's a big woody one where they're just at maturity and was preserved. So I just want to show you the range of variation. Either side here is from Sucker Creek. The one in between is from Paddock Reservoir. In the rose family, there's quite a bit of diversity of things, but we have a serviceberry on the left. Over here on the right, we have Toyon, or Heteromeles is the genus. And down below in the center is even meadow sweets, Spirea. There are cherries, prunus of various kinds, whether they're almonds, cherries, you name it. There are rose family, rose proper things. There's a rose hip. There's rose leaflets, even a raspberry with a couple of leaflets attached to a big thorny peduncle, or excuse me, yeah, petiole. There's tilia or basswood, and the leaves tend to be pretty delicate, and also the insects seem to find them delicious. So they often get chewed up before they ever get preserved, but every once in a while you find them. But far more resistant and apparently less tasty are the bracts. And so here's a bract, and they're distinctive for having that peduncle and a fruit or more at the end. I wish a little bit more of this had been preserved, but you take what you get. There's also a bunch, maybe 5, 6 kinds of poplars, cottonwoods, and aspens that we have. I just put in one to remind me that, oh yeah, we have those. Same with willows. There's lots of willows. These two are very similar, but I think they can be told apart by the prominent teeth versus very subtle teeth and an angle of the secondary veins departing and several other characters. But there's also broad, much broader willows, little short, round, almost orbicular willows. There's quite a range of willows in the Miocene of Idaho. There's at least 10 species of maple. Ironically, all of them are known from Sucker Creek. It's the Flora with the most different kinds of maples that's ever been identified by people who revised all the maples. And I just put in 2. There's a red maple type on the left and a silver maple type on the right. Here's some Samara range of variation where that straight line is the suture where the other paired seed was. Down here, it's a little narrow tip. Here, it's a fairly broad edge, etc. So there's lots of variation in the wing seeds, the Samaras of maple. Then we get back to that Trochodendraceae family I promised we'd get back to. And this has been called many things, but is currently settled on Zisophoides. And it appears to be an extinct member in this Trochodendraceae. And the leaf is incredibly variable. Lots of different leaf morphologies from what we think are juvenile leaves all the way up to mature adult leaves. And it's kind of fun. And then almost always co-occurring are these 2 fruits, this kind of fruit on the left, and it's called Nordenskioldea. It was named separately and never found attached, so they still leave it as a separate name, separate genus. But it is roughly, I'm making it up, but I'd say 80 to 90% of the time it co-occurs with Zisophoides. And then rarely we'll find Tetracintron, a modern genus, in the Trochodendraceae, and we can even find the fruits, the infructescence, the collection of all the seeds. Then there's Trochodendron proper in the Trochodendraceae, and this is the seed capsule from Sucker Creek, and on the right is a specimen of a modern individual just for comparison. There are ashes, Fraxinus, and here's a fun one because it's a compound leaf and you could see 2 leaflets attached with the asymmetrical leaf base and the irregularly spaced and shaped teeth. That's Fraxinus. And then there are lots of Samaras, perhaps 4 or 5 different kinds of Samaras, winged fruits that are found here. One of which was from Horseshoe Bend, and they bulldozed the site away. I was really bummed when I came back one year and it wasn't there anymore. The highway department took it away. If you know old Suicide Curve, as you go north into Horseshoe Bend, it isn't there anymore, but it had some neat fossils in it. In the elm family, we have several species of elm, but this one's like the American elm. It's called Ulmus americana, but it's just like the common elm that's around. And then there's this thing called Zelkova. Modern name is the same as the genus, just like hydrangea, but it's an elm-like plant that almost never ever has secondary teeth on the teeth, little side teeth, while regular ulmus almost always has secondary teeth. And then there are several other characters that identify this as Zelkova. Monocots, I just want to show you a couple really quick. This is a fun one called Caldesia. This is an incomplete, but a leaf of an aquatic plant. And then here on the right is sedge or carex. And carex is fun. I showed this to a world's expert on sedges, and he said, yep, there's no question it's carex. And if it's one of two sections in carex, and if I'd get him the fruit, he would tell me what genus or what subsection it is. But anyway, no question that it's sedge. So that's kind of fun. We had monocots. There's also assorted grasses and things. And then I think this is the last mega fossil I wanted to show you. And this is an odd seed. We just don't know what it is. It reminds me of an almond because of this texture. And so I think it has affinities to the Rosaceae, the rose family. But it because there's also, if you look at a raspberry seed up really close on the magnification, same pattern. And then what's famous about it is, to me, or interesting about it, it's found only in 3 areas in Idaho and nowhere else on Earth. So the Sucker Creek area straddles the Oregon-Idaho border, the Ponderosa Flora, and then up in Clarkia, both the racetrack and Emerald Creek. It turns out we've got 30 or 40 specimens from it in McCall area. So we have a lot of it to stare at, and it's just a mystery. If you ever want to share with me what you think it might be, I'd love suggestions. Okay, pollen, I have 3 slides to show you just to show some diversity of it, but a lot of these genera are going to start to sound familiar. Over in the upper left is spruce, then pine, and letter C is hemlock. D and F are in 3 families that are in the redwood, greater redwood group. They call it the TCT group. E is larch or Douglas fir, both in the pine family. G is Podocarpaceae, a southern hemisphere thing, or Cathea, which is a monotypic, a single species genus in the pine family. And we frequently find this in these floras, never abundant, but always regular. It's always there. So Podocarpaceae, and then there's a cedar in H, and there's a fir in I. If we jump up to the lower vascular things and then on into the angiosperms, we see a lycopodium or ground pine in A, an unknown spore in C is Selaginella, a spike moss. In D and F are 2 types of ferns, a wood fern type and a polypody type, and another spore we don't quite know what is. There's also Potamogeton pondweed. H is Typha or cattail. I is some kind of grass family. They're always characterized by a single little belly button looking pore. J is birch, K is alders, L is elm, M is in the 2 families that have been lumped together, Chenopodiaceae, Amaranthaceae, and this happens to be the goosefoot type, more like the old amaranth subset. N is high spine compositae or Asteraceae, sunflower dandelion family. You find them with wicked spines or very subtle spines. This one happens to be one of the big wicked spine types or high spine comps. And there's even lily pollen. And then finally, all of these should start to sound familiar. Hickories, Turoccaria, which is an Asian hickory called wingnut, walnut, juglans. D is liquid ambar, E is willow, F is maple or acer. G is ilex or holly, H is oak, I is ash, J is beech, K is a tetrad, a collection of 4 pollen grains in the heath family, and then L is Nyssa or Tupelo. I didn't happen to show you one of those, but we have those as leaves and as well, and seeds. Finally, there's wood. So we have wood in various places. And wood anatomists take 3 distinct slices to identify their wood. So they use a cross section, sometimes called a transverse section. They use a radial section like a radius or a spoke on a wheel or a cut in a pie piece. And then at right angles to both, they use a tangential section. On the right-hand side are 3 cartoons of a cherry from Sucker Creek, fossil prunus, with those 3, a cross section, radial section, and tangential section. And then if we look down below on the right, we see those same 3 views, multiple images in some cases of cross sections, radial sections, or excuse me, tangential sections and a radial section. On the left are 3 images of an oak, various oaks with patterns showing this springwood, summerwood, springwood, summerwood pattern. So I thought those were kind of fun to put in here. We also see ginkgos. Ginkgo leaves preserve. Ginkgo pollen is very tough to preserve, so you almost never see it. And there's Aesculus or buckeye, and there are a few leaves of that as well. So concluding, winding up here, what can you do with all this stuff? Well, they found out way back in 1916 and 1918 that if you look at a leaf and just determine are the margins smooth or entire margin versus tooth for dicotyledonous angiosperm leaves, it turns out there's an exact linear relationship between the percent of those entire margins and the mean annual temperature, the temperature under which they grew. And since we know that temperatures decrease with increasing elevation and with increasing latitude, then we can also estimate moisture, available moisture during the growing season based on percent entire margins. We can also compare this, here's percent entire margins through geologic time and also with temperature on the right. And what we see is a lot warmer but fluctuating temperatures throughout the first half of the tertiary. And by the time you get to the second half, it's fluctuating but much cooler. And the V happens to be centered right on where Sucker Creek would be. So this 15 million year interval I'm talking about. We also can compare these fossils to what we think is their most similar living descendant. And we can compare how many of those need or require summer rain. So this is a way to assess the Mediterranean climates in Western North America. And if we rank them before 15 million and after 15 million from north to south, what we see is there's a pretty good correlation. And at that time, Sucker Creek was considered right at 15. We now suspect it's probably just a smidge older, closer to the 16 million. But this was something that people have been queuing in on lately called the mid-Miocene climactic optimum. So that's a whole other talk, but I just wanted to show you that it does tie in. And then lastly, believe it or not, there's a lastly, we can look at a sequence of rocks, and this is the one in the greater Owyhee region, the composite stratigraphic section. And down here, we see the Sucker Creek Formation. And if we compare that over to the right to the type section, the sequence of rocks that defines the Sucker Creek Formation. And if we look, all these little tick marks are places that have been sampled for pollen, and they process the pollen. And here's a little blown up piece of a pollen diagram just to show you, and I made it really big, so it's kind of fuzzy. I'm sorry for that. But certain types like fir and spruce indicate upland, mountainous regions. Certain types like pinus are found throughout, but more common in lowland or warmer regions. So we can see in this case, there was an abrupt change that happened right at this point in the sequence. If we compare that back to this type section, you can see that we can reconstruct what the vegetation was like at various intervals along here based on what the pollen is telling us up one hillside. And that's a neat way to do this. And what we've noticed is, and this is the greater we, not me, is actually, these ones were done by Ralph Taggart and Ariel Cross, my major professor and his major professor. But we see that there's the immediate post-disturbance phase where there's a few relictual pines and then just a few little plants starting to come back after some kind of volcanism or something. Then we see the pine parkland stage where pines become established and you get more broadleaves down low by the water, but generally it's dominated by gymnosperms. And then up here in the upper right, you get a maturing, but not yet mature forest that's mixed, and the pines drop out of the lower elevations, but are persistent in the upper, the gymnosperms. And then finally, we get a mature closed canopy forest. And below them are written the genera that were found that justify this. Here's a close-up of the most mature one, because there's the most fun things here. And so montane conifer section is characterized by fir and spruce. The mixed pine, mixed hardwoods are characterized by elms, oaks, birch, alder, pines, barberry, that Mahonia, hemlock, poplars, oaks, and vaccinium. Then we come down to the bottomland slope and hardwood forests, and we see that just basically the gymnosperms drop out. And then as we get closer and closer to water, you get the alders and willows starting to dominate. And then finally, with their feet wet, as I say, you get that Glyptostrobus and bald cypress and cattails. So that's what we can do with these things. So the last point I want to make very quickly is there any modern analog to what Idaho's forests were like 15 million years ago? And I'd say start with a bit of a modern Chinese mixed broadleaf forest, such as Chiangbai province in China, which has a pretty good preservation of a lot of nice diversity. Add to it a touch of southeastern US aquatic habitats, marshes, swamps, that sort of thing. And then add to it a touch of mountains, like plants on mountains like you'd see in the Klamath Mountains in northwestern California. And all of those might represent the composite that we see. And then mix in a good dose of volcanic disturbance. So we have lavas that flow and dam up the valleys. We have volcanoes that blast off and cause disturbance, but then silt in. And so our fossils get preserved. And then we have a slow recovery of all these things over time as the vegetation recovers. So thank you very much. And I'm open to any questions people might have.
Thank you so much, Pat. I want to say that if we were all in a room together, there would be just enormous applause. That was extremely fascinating and intriguing for me to listen to. And I really loved the backdrop that you gave of the geological time. And then you mixed in all the characteristics of plant morphology and how plants have gone through the Miocene time to what we might currently think of was happening back then. And I was just enthralled the whole time. I just got to say.
Well, thank you so much.